Hi Science 10, welcome to the video lesson on how materials are transported across the cell membrane. So remember we have this phospholipid bilayer structure and embedded proteins in the cell membrane with the molecules that may be attached to them for cell recognition. So the first term here, Brownian motion, means that there is a constant random motion of particles. And so as you can see, if you follow one particle to position 1, it'll just keep bouncing around and around. And that's because whenever it hits another molecule, it will bounce off. So it's got that kinetic energy that keeps it moving. And so because molecules have Brownian motion and will continually bump into other molecules and move, that is also the reason that molecules will move from an area of high concentration to area of low concentration. So where there are more molecules, they will naturally move to where there are fewer molecules. And that's a fundamental concept of all of our methods of transport for this topic. Concentration gradient means that there is a difference in the concentration of one area compared to another area, and you will see that movement of molecules from the high concentration area to the low concentration area. So that is all what it means by molecules will move down the concentration gradient. It means they will go from high to low concentration until they reach equilibrium, which means until they are equally as concentrated in both areas. That will be when it's equal, it's in equilibrium. So the molecules at equilibrium, when they do become equal on both sides, it doesn't mean that they will stop moving altogether and be still, because they're still going to have particles to bump into. But what it means is if one goes this way, one goes this way, they will continue to be found in equal amounts on both sides of the gradient. So the direction of particles, movement of these particles, pretty obviously, because there's a higher concentration on this side, lower concentration on this side, the molecules will move from high to low until they are equal. So sometimes when it's just passive transport, it means it naturally goes from high to low. Doesn't have any energy needed for that. Sometimes if there is movement from in the other direction, so where there is very little and the cell wants to take in more, that would require ATP energy, and that's one called active transport, because it'll actively take energy to move that molecule over there. So we're going to examine all these different kinds of movement of molecules. And the first one is easy, passive transport. These are types that do not involve any ATP energy, and these are types where it will always go from high to low concentration down the concentration gradient. So the first one is diffusion. You've heard that before for sure in grade 7 and 8. And diffusion is just when particles go from where they're high to where they're low concentration, and there's no energy required. So if you put a drop of dye in one container of water, you'll see that eventually it will move to where there is no dye until it's equally concentrated all throughout the entire container. And that's an example of diffusion. So if you've got the red guys on this side here, and they're allowed to move through that membrane, they will move through until there is the equal amount of red molecules on both sides of the container. So there's the dye drop example, and here's the similar picture with the yellow or the orange molecules getting dissolved. So if you have like a sugar cube, solute like sugar put into water, it'll dissolve until it's equally the same amount of sugar out everywhere. That's all pictures of diffusion. Okay, oxygen, it says, is an interesting one, because when they move through into the cell and through the phospholipid bilayer, it's going to be used up very quickly by the mitochondria doing cellular respiration and using up the oxygen. And so what happens is they're continually, as the blood flows by, continually has a higher concentration of oxygen that it got from you breathing it in through your lungs. So as the blood goes by cells, it will continually be moving that oxygen into the cell. So if you can imagine the oxygen being outside the cell, that's what extracellular fluid is. It's going to be simply diffused in through this cell membrane and into the cell. And so therefore it says what the concept here is saying, that it will never reach equilibrium, because it would continually, it'll go in and it'll get used. So there's never going to be an equilibrium for oxygen because of it getting used up all the time. So it's continually going to be coming in. Okay. Number 2, osmosis. All that is it is still diffusion, but it's diffusion specifically of water. So when water goes from where there's lots to where there's less, that is osmosis. So simply you can take a dry face cloth and dip it into water, and water gets soaked up into that face cloth. That is osmosis, because water goes from where there is lots to where there is less. So water being the blue molecule here, you'll see that it's going to always try to move to where there's less water. Another way to look at this is that it's always going to try to dilute more concentrated areas. So water is known to go into the sugar, you can think of it that way, to try dilute the highly concentrated sugar zone. The word tonicity means how concentrated it is. When you compare something that's hypertonic, it's more concentrated with solute, and hypotonic, it's less concentrated with solute, and hypo, meaning water, means if it's less concentrated, it also has more water. So here we have a picture of a red blood cell that is normally looking like this, kind of has a dip in the middle. And when it's in isotonic, or fluid that is the same concentration as that blood cell, it'll happily function in its normal size and shape. And so if you think of blood or even Ringer solution that's used in an IV fluid, those would be isotonic to the blood, and they would keep the blood cells basically in their normal size. If you put that blood cell in a hypertonic, if you think hyper, thinking lots of sugar, if you put that in a hypertonic solution that's got lots of sugar or lots of salt, then what's going to happen is that the water in the cell is going to try to dilute the hypertonic solution, which is highly sugar or highly salty, and it's going to shrink it. So you should know that that is going to be the result. If you then put the same cell in a hypotonic solution, that's basically like entirely water, less solute is hypotonic, and so this means that now this cell is basically more concentrated than the water around it is, because the water has no solute at all. So water is going to go try to dilute that cell this time, and it will swell the cell. Okay, that's the 3 tonicities that you have to know about. There's actual diagrams of an isotonic red blood cell, one that's in a hypertonic solution, so it's so salty on the outside, for example, that that it's going to have any cellular water try to go dilute that salt and shrink the cell. And a cell in, I got this wrong, I see that, okay, I see that now. This is our isotonic, I was wondering if why that looked wrong, pardon me. Okay, this is the isotonic cell, red blood cell that is same concentration inside and out, so this is actually the normal cell in this diagram, and this one is the one that's put into pure distilled water, and that's hypotonic, and that would be like distilled water. and therefore the water will try go in and dilute our cells and swell those cells as a result. So this shows you the exact same thing, except it shows you what happens in a plant cell. In a plant cell, if you put it in a hypotonic solution and water goes in, it'll actually it will fill the big vacuole and make it the cell nice and plump. So watering your plant will do that good thing. In an isotonic solution, water may kind of leave as well, and it says that might slump the plant a little bit. And then in a hypertonic solution, if it's like in salt water, what's going to happen is that cellular water will go out and try dilute the salt around it and shrink back. The cell membrane here has shrunk back, and it'll get like ultra floppy and droop over. Okay, so this one says that the water is being transported into the cell, which means that this must have been a hypotonic solution that it's in. That's my guess. Water is going to go in and try dilute the cell. This one is one where water has totally shrunken back the cell membrane here. So this must be a hypertonic solution, and water all left to try dilute the hypertonic solution around it. And in living cells, too much or too little water can kill a cell. So the cells in a healthy situation are always surrounded by isotonic fluid. It says if plants are in hypertonic solutions, they will wilt like salty or sugary water. And if they're put in hypotonic solutions, they generally won't actually explode because they're so full, because they have the cell wall that will stop that. But they will totally plump up compared to cells in sugar or salt water. You can see how the cell membrane pulls in and they're shrinking back. Okay, turgor pressure. When that vacuole is really full, it keeps that cell nice and firm and rigid. And if an animal cell does shrivel up, it's also called plasmolysis, it could actually die. And you guys can check out this video about the cell being represented by an egg. And this demo with potato slices is one where we took a mass of those potatoes after they were put in different tonicities of water, and basically the one that was salt water, it was so concentrated that the fluid came out of the salt water and came out of the potato and into the salt water, and your potato totally shrunk up and became the smallest one. Just kind of what happened. And in distilled water, the potato got all plumped up and was the biggest one in mass. So those are masses that were taken. Okay, so next kind, we did diffusion and osmosis. Next one to talk about is facilitated diffusion, and that just means that there's a protein that helps move the molecules across the membrane. And it says it's still diffusion, so it's always still a material that's going from high concentration to low, but there's just this protein here, like this carrier protein, that takes that substance and moves it into the cell more easily. So they say that if it's supposed to go across, if it's got the carrier protein, it'll go way faster than if you just wait for the diffusion to happen naturally. So it's still going from high to low concentration. And sometimes besides carrier proteins, there can be a channel protein that's just got the tunnel through it that allows certain materials through. Okay, here is a facilitated diffusion example where we've got molecules inside. There's more of them, so they will move outside, but they've got this carrier protein that's helping to move them. This is showing a sodium-potassium pump, just that there's carrier proteins that will move sodium and potassium in and out of the cell, and that's specific to the nerve cells in the body. When impulses go down nerve cells, sodium-potassium pump has to happen in order for that to be completed. Okay, here's a channel protein, just has a tunnel through it, whereas a carrier protein actually grabs it and pulls it across. You can check out the video animation on that. Okay, more of the same picture. All right, next one we're going to talk about is active transport. And this one is different because your cells are actually going to be grabbing more of something that they already have a lot of. And so it's going to take energy to pull more in when there's already more higher concentration inside. And the example I like to talk about here is the iodine in your body. Any iodine in your bloodstream will be grabbed by your thyroid. That's found in your just above your larynx in your neck, and your thyroid gland needs the iodine to make thyroxine hormone. So they say there's a 25 times greater amount of concentration of iodine in your thyroid than anywhere else, but it continually actively transports it in because it needs it as a building block to make the hormone. So what happens here is ATP is used, and also a carrier protein is used, both of those, to get materials from where there's less to where there's already lots. So in the comparison of these three processes, diffusion is just high to low, facilitate diffusion high to low using a carrier, and active transport is going from low to where there's already a high concentration, but that is a lot of work to grab it in where there's already a lot, so ATP energy is necessary. So you guys have to know these three processes and how they compare. Active transport showing you that ATP is used and breaks down into ADP when it's used. And they mention the sodium-potassium pump for nerve impulses, how that's used. You can see an animation on that here. Plant roots, they use active transport to move minerals out of the soil into the cells. So they continually grab minerals in, and that's going to be done by active transport as well. The lab that we would have done with yeast cells is that there's a dye, a red dye that's put into the yeast sample, and some of the yeast samples are alive and some are dead. And the ones that are dead stay red inside. I guess I should use red to show you that. They stay red inside because they can no longer, because they're dead cells, no longer do the active transport and pump them out, pump out that dye, because it's all floating in the red dye as well. But the cells that are alive, they can actually pump that red out, and that would be done by active transport, and you end up seeing them under the microscope as clear living cells because they actively transported out the dye. Okay, the last two things, endocytosis, exocytosis, are processes used to get large molecules in and out of the cell membrane by basically making a vesicle and folding in. So endocytosis is when your membrane folds in and forms a like a vacuole or vesicle by sucking something in. And exocytosis is when the same thing happens by taking a vacuole and basically spitting it out. Endocytosis can be done with large objects, which is called phagocytosis, and done with like liquids, which is like cell drinking, and that's called pinocytosis. So here is phago taking in a large molecule and sucking it in, making a vacuole, like this picture. And it can also be done with liquids, and that would be pinocytosis. The opposite, exocytosis, is when the cell is trying to get rid of something. It could be waste, or it could be something it made, like a hormone, and it would fuse its vacuole or vesicle membrane to the outer membrane, cell membrane of the cell, and basically spit it out and release that. So exit means leaving, so that's exocytosis. Bringing it in is endocytosis. And there's an actual picture of it there. Alrighty. If it's receptor-mediated endocytosis, then it would be brought in because of a specific protein molecule that's recognized. So sometimes there is a protein involved in it. Both of these do involve ATP energy. It takes energy to fuse the vacuole and bring it in, or fuse the vacuole to the membrane and send it out. So those do use ATP energy to happen. Two examples of membranes. One is reverse osmosis, where we've got water being pushed through a membrane, and basically pure water can get through, but bacteria or other impurities do not. So water is pushed through this membrane and ends up being pure water on the other side. And there are like big jugs of water you can buy or get a process in your house that does reverse osmosis. And then kidney dialysis is where someone's kidneys don't work, so they have to filter their blood. Instead of doing it through their kidneys, they have to go in to be hooked up to a dialysis machine and get the impurities and toxins in their blood filtered out by their blood going through this tube here, but there is fresh dialysate fluid around it, so things that are toxic in the blood naturally diffuse out into the toxins diffuse out into the dialysis solution, which is then collected, and their blood goes back without the toxins, goes back into their body. And this picture just shows you their blood cells are too big. They don't go out, so the blood cells stay and go back to the person. But these bad guys here, the yellow ones with the X on it, that would be bad things your blood has in it as wastes, like urea and uric acid and ammonia. And because there's more of them in the blood going into this machine, they would diffuse out. And that's how dialysis works. And so the person is getting this pure blood back into their body without those toxins in it. And that's a picture of a dialysis machine in the hospital. Alrighty, we are done with chapter 8, folks.